Good morning, everybody. I'm very happy to introduce today's speaker, Dr. Goutam Charkraborty, who is also a dear friend and colleague. Gautam did his PhD at National Center for Cell Sciences in India, following which he worked as a postdoctoral fellow at Memorial Sloan Kettering in New York. He continued working as a junior faculty in Memorial Sloan Kettering for a few years, and a couple of years back, he started his own lab and is currently an independent PI at Mount Sinai Icahn School of Medicine. Gautam's been very successful in his short career. He's published a lot of high-impact papers, and he's got funding from the Prostate Cancer Foundation, the Department of Defense, as well as an R01 from the NCI recently. His work is primarily focused on BRCA1, BRCA2, and prostate cancer. So welcome, Gautam, and I'll let you go on with your talk.
I say thank you so much for this kind introduction. Hi all, good morning. As Sid mentioned, I'm Goutam Charkraborty. Currently, I'm an assistant professor in Icahn School of Medicine, Mount Sinai. I'm affiliated in Department of Urology, so that you understand my research is mainly focused on genitourinary cancer. As a basic scientist in a part of the clinical department, my goal is to understand what happened in the clinic. As I said, on that my research focuses on bedside to bed, means we try to understand. Wait for this one, yeah. This is the goal of my lab research focus. What we do actually, we try to understand what happened in the clinic and try to replicate the result in the particular genetic mutation in our lab system. From the result of the mutation, we went to find like that what will be how this particular mutation or genetic alteration affect tumor growth in mouse and how can we treat them. And then we try to collaborate with the clinician, although I'm not still very much successful yet, but we are continuously trying to do that. So bring our bench research to clinic to start a clinical trial. So this is our area where we do the research and try to find a cure for the cancer, which is mainly focused on prostate cancer. So why prostate cancer? So prostate cancer, prostate gland is a male gland, it's a male sex gland, and it's a small gland in the body. Means even if you remove the gland, it's not affect except sexual function. But if the cancer happen in prostate, particularly then most of the time the cancer is indolent disease, and if they detect early, they can be curable by surgery. But once this cancer leave the boundary of the prostate and metastasize in different part of the body like bone, brain, or liver, they're very lethal. So prostate cancer is happen in 1/6 of men in the over 60, particularly incidence is significantly higher in the African American men and the veteran population. So it's a very high burden for the veteran and also the old population of this country, not only United States, but all over the world. The several years, particularly like almost like 100 years ago, first time detected that prostate cancer is responsive to the anti-androgen therapy, and that is the mainstay of prostate cancer research. Last 10 years, there is significant improvement happened to find the treatment, but where we are now. So I always show this slide to show that which is our current situation. This is the prostate cancer statistics from 2010 to 2023. As you can see that there is not any significant improvement happened to the treatment and increase the survival chances of the prostate cancer. Even after 10 years, there is almost 13 years, significant improvement of several new therapies, including immune therapy, lots of kind of high level of sequencing studies, but we still 34,000 or 11% of the men are dying in prostate cancer, which is very significant and very concerning. So what we are doing, what we are missing, that is the starting point of my lab research. So if a patient have a prostate cancer or any kind of discomfort in the prostate, they come to the clinic. And currently I'm working in the urology department. I talk with the urologist to understand that what is the first step it can be done for when a patient have something discomfort in prostate cancer, particularly if they have a high rise of PSA or cancer is detected by the biopsy or MRI. So what I understand with talking with them last 2 years with our urologist, my department, there is actually 5 options there. You can tell either they are going no treatment like active surveillance or watching when the cancer is inside the boundary of the prostate and very low grade, lesser than Gleason 6 or just the beginning, but Gleason 6 disease 3+3 or 3+2. If the cancer is like Gleason 7 or Gleason 8, there is a chance of either prostatectomy or radiation therapy. The concern, what our departmental urologists say, that they don't have any kind of biological marker where you can detect the patient can go to prostatectomy or radiation therapy. And some of the patients I mentioned that, which is the mainstay of prostate cancer treatment, which is the hormonal therapy or anti-androgen therapy, all the patients after prostatectomy, which have the aggressive or symptomatic disease, undergoing hormonal therapy. And some patients, even in the neoadjuvant setting, have undergo hormonal therapy, but this is not that much successful yet. So the question is like that, we don't have any genetic marker to decide our decision of how do we treatment the prostate cancer. So if we see the genetics of the localized prostate cancer, there is 60% of the prostate cancer of ETS transcription factor fusion, TMPRSR or TMPRS ETB1. 10% of the prostate cancer have a SPOP, which is one of the DNA damage pathway gene mutation. Around 20% of the prostate cancer have PTEN or PI3 kinase pathway alteration, particularly mutation of, particularly deletion of PTEN, and other genetic cause happening around 10% of the prostate cancer. But neither of these genetic information can change the decision what made by the urologist. So we still don't know how can we detect indolent to aggressive prostate cancer and tailoring the treatment. It can be going to a prostatectomy, it can be going to a surgery, or we can give other kind of drugs. So that question asked me when we see this question, and I was, before that I worked in the Department of Medicine in Memorial and worked with Dr. Phil Cantor, when we are talking several times that how to move forward. So if we can see the prostate cancer treatment nowadays, because there is last 10 or 15 years, there's several treatments are introduced for the prostate cancer, particularly second generation anti-androgen therapy, chemotherapy, PARP inhibitor, which is, I will talk lots today, immune therapy and radiotherapy. But all the treatments are for the latter stage of the disease, which is called metastatic castration-resistant prostate cancer. But this kind of treatment actually work for 2 or 3 years for the pain, and then every disease eventually gets resistance. Therefore, currently, we think that option for us, a current window of opportunity, if we can tailoring the treatment in the early stage of the disease to know early stage of the disease. To knowing early stage of the disease, we need to know what mutation actually enrich in the metastatic disease that may be transform the indolent disease to a metastatic castration-resistant prostate cancer. Few years ago, our leaders had come together that time, including my mentor, Dr. Cantor, and others in the United States, and they formed the dream Stand Up to Cancer Prostate Cancer Dream Team. What they did, they collect around 500 metastatic castration-resistant prostate cancer and sequence them to understand their genetic changes in the metastatic disease. The result is quite surprising because there is some mutation are showing in the prostate cancer. This is the whole map of the genetic mutation, what happened in the metastatic castration-resistant prostate cancer. This paper was published in 2015. One of the important part of the mutation, they found the mutation happening in the DNA PI3 kinase pathway, which is already mentioned, which is kind of known, but the other mutations happen in the DNA damage repair pathway, particularly a significant amount of the prostate cancer patients, which is previously not known, have BRCA2 mutation. But another part, we also see there is a significant changes in the copy number changes in the prostate cancer, like copy number changes in these metastatic castration-resistant prostate cancer cases. What is copy number changes? The copy number is the somatic change in the chromosome structure that result gain or loss of the DNA. What we do see, the metastatic castration-resistant prostate cancer have a high burden of the copy number changes. And it's also known that the copy number changes is associated with lethal disease, not only in prostate cancer, but in several other cancers. But it is a copy number change is just a random event or specific part of the chromosome get changed. To do so, when I was in Memorial, so we used the Memorial Sloan Kettering Cancer Center dataset, which is called MSK Impact dataset. There is like a fraction of primary and metastatic cancer spaces, prostate cancer samples are sequenced in a single institute. And that sequence is particularly focused on specific gene, like a 423 gene, but also they are also check the copy number changes using a facet algorithm. We collaborated with the Dr. Nikki Swartz group and Subi, who is the bioinformatics engineer, did an amazing job to overlap the copy number changes primary versus metastatic disease. As you can see, the primary prostate cancer actually is the green color, and the metastatic prostate cancer, which is the violet color. The surprising for us, we found every chromosome in our body get amplified in the metastatic disease compared to primary disease, which means that a gene amplification is not a specific event, it's kind of a random event. This is interesting for the X chromosome, which is actually get amplified, and X chromosome also carry the androgen receptor, which is a major molecule which drives the prostate cancer. But if we look the other side of the genome, means the part which is deleted, there is only specific areas which are deleted, which are enriched in metastatic disease compared to primary disease. We get a cutoff mark of 25% and significant value. What we found that the areas are deleted are significantly restricted, means if you compare primary versus metastatic disease. The areas which are mainly deleted is the chromosome 10q, which is the known gene P10 I mentioned, which is long time actually known. But surprisingly, we found there is a significant whole chromosomal deletion in chromosome 13. And the area we'll talk today, it is actually two gene which is deleted called BRCA2, and then his chromosomal 9, but RB1. Importantly, we also found a significant deletion in the chromosome 5q. The gene which we found that which is mainly deleted, there is only one peak, as you can see here, that is like PIK3R1. We have several work on PI3K1, although I am not talking this PI3K1 today. But as you can see, this map have a missing part. We don't consider here the male chromosome, the Y chromosome, which is the other second part of my talk today, is what is the role of the Y chromosome in cancer. So now we'll talk first part in the what is the impact of chromosomal BRCA2 and RB1 deletion in prostate cancer, and how this deletion may transform the indolent disease to metastatic prostate cancer. So BRCA2 RB1 deletion is pretty unique because of this chromosome 13 is a little unique chromosome in our body. It's the largest acrocentric chromosome in the body, means this chromosome only have the Q arm, no P arm. But what we found like that, this chromosome actually mostly deleted in the prostate cancer. And this data actually known for like 30 years before, 1990s, and that time when our cytogenetic researchers used the karyotyping of the prostate cancer tissue. We do know that time this 13q is deleted. Again, the sequencing study also. Hello. Hi. Hello. Sorry. The chromosome 13q is deleted, and we can show that this 13q deletion actually frequently found in the prostate cancer patient, significant amount of when localized prostate cancer patient. So our first experimental direction is what is the role of this 13q, particularly BRCA2 RB1 deletion in prostate cancer, and can this deletion just a passenger event or is a driver event of the prostate cancer progression? To do so, what we did, this BRCA and RB1 is a two different gene actually, even they are chromosomal level. BRCA2 is mainly regulate the homologous recombination repair, replication, Fox stability, or DNA damage pathway. Whereas the RB1 associated with the cell cycle progression, genomic stability, or DNA damage repair pathway, partially. But these genes don't have some very much overlapping function. So when we map this thing in the TCGA prostate cancer samples, we do set the patient who have the BRCA2 RB1 co-deletion, because of most of the time you can see BRCA2 is deleted with RB1. There is a very few sample where is BRCA2 only deleted, not with RB1. And there is a fraction of the patients who have only RB1 deletion, not the BRCA2 deletion. We consider both copy number loss, means like a single copy loss and double copy loss together, because we do found loss of BRCA2 even in the single copy significantly downregulated the protein expression of BRCA2 in the clinical samples. What is when do a survival curve? We found that this is the red is the co-deletion, and these co-deletion patients have a lesser or poor prognosis compared to patients who have no deletion or the actually deletion of the RB1 only. This is one of the interesting part we found, that if the patient have a BRCA2 RB1 deletion that is enriched in metastatic disease, almost 50% of the metastatic disease have the BRCA2 RB1 co-deletion. But importantly, if we find even a very less number of patients, even in Gleason 6 disease, have this BRCA2 RB1 co-deletion. Important part is like that general Gleason 6 disease is a very early stage with the prostate cancer. They are not very aggressive, and most of the time they don't need even surgery. But we do see from some of the data set around 10% of the Gleason 6 disease can evolve in the very aggressive disease. And we don't know this, which is the 10% of the localized Gleason 6 disease who are transformed to aggressive disease have BRCA2 RB1 co-deletion or not. But if I see this graph, we can understand that BRCA2 RB1 co-deletion enriched along with the Gleason stage, particularly Gleason 8, which is that known as a localized very aggressive disease, have almost 40% of BRCA2 RB1 co-deletion. So the experimental process, we take the LNCAP cells, which is a common cell line we all prostate cancer researchers use. I like to mention that unlike many other tumor types, prostate cancer have very restricted number of cancer cell lines. We have only 7 cell lines out of only 3 or 4 cell lines, which is actually have expressed androgen receptor and sensitive to androgen signaling pathway. So that they are the major, they're the major cell line we use for our research purpose. So we first use the BRCA2 CRISPR and then knock down, and then use the RB1 shRNA to knock down BRCA2 RB1 to make the BRCA2 RB1 null cells. And then we analyze this, we do RNA sequencing study. The RNA sequencing study show that there is a stepwise changes of the gene expression. As you can see, when you knock down BRCA and RB1 together, we found like that there is a significant changes of the gene expression compared to one gene alone knockdown. We found these cells which are knocked down of BRCA2 RB1. We do see that there is a changes of cellular morphology, which kind of associated with the high invasion and maybe epithelial to mesenchymal transition. We did some downstream analysis, and we found one of the molecule which upregulated in the BRCA2 RB1 loss is SLUG, which is another known EMT transcription factor. And do observe that if we knock down SARP with even siRNA in the BRCA2 RB1 knockdown cells, there is a significant downregulation of the cellular invasion, indicated that BRCA2 RB1 loss mediated SARP activation leads to epithelial to mesenchymal and invasive phenotype. But what is the molecular mechanism behind it? But before that, I want to show that this paper we published in 2020, after that several groups actually reanalyzed the data, and it found that our analysis actually and we found in several other cohorts. Like this paper is published from the Japan, where they found one of the prostate cancer patients who have the very early stage of the disease, but have a BRCA2 RB1 co-loss. And they suggest that such kind of patient, even if you found in the early stage of the disease, you can surgically remove them. And after this surgically remove this patient, the cancer is like 2 years done, and the cancer never, there is no what we call the biochemical recurrence biochemical recurrence or PSA activation in the PSA found in the bloodstream. So means if you can found this disease very early stage, you can surgically remove. Actually this group suggests that if surgically remove this, and then patient can be survive better. The result from Spanish group, they show that also these patients who have the BRCA2 germline carrier mutation or BRCA2 germline carrier plus BRCA2 RB1 co-deletion, they are more aggressive than BRCA2 germline only. So means BRCA2 induced aggressive phenotype actually driven by co-loss of RB1. And very recently, this paper from the evidence group in the Harvard, they showed that the patients who have more immunogenic tumor have a very high co-loss of BRCA2 and RB1 compared to the non-immunogenic prostate cancer, indicated that the phenotype or the subtype of the prostate cancer we demonstrate actually are found in several other datasets, and they may associate it with a bad prognosis. But we need to know, if it's a bad prognosis, what is the treatment option? The question is what I first time raised, that if this patient do not respond to a common therapy like anti-androgen therapy, which is coming later, because we do see that BRCA2 RB1 loss leads to the resistance to anti-androgen therapy. But what could the better option? So BRCA2 when the BRCA2 null cells or did any kind of DNA damage repair deficient cells responds to PARP inhibitor-based synthetic lethality. And common action of PARP, which is like a regulate the single stranded DNA repair pathway. If the loss of BRCA2 or any kind of DNA damage repair pathway, cells are dependent on single strand DNA repair. But if you block the PARP, which induce the synthetic lethality, means the whole pathways are dependent, whole two pathways are dependent to interaction between two genes. One gene is not the whole pathway, whole survival pathway depends on only one gene, which is PARP. And if you block the PARP, cells can not survive anymore. This is a basic concept of synthetic lethality. Although we completely don't know how PARP actually works, but in clinic, we do see PARP inhibitor working, not only in prostate cancer, is very good successful in the breast and ovarian cancer, the people who carry germline mutation of BRCA1 or BRCA2. We do see that PARP inhibitor in the recent time in the prostate cancer, particularly one inhibitor called talazoparib, they significantly induce survival of the prostate cancer patient who have the BRCA2 germline, BRCA1, BRCA2 mutation, and this when the talazoparib combined with enzalutamide. Importantly, even the talazoparib combined with enzalutamide, which is an anti-androgen inhibitor, it also induced survival, means like a better survival of the patient who don't have known BRCA2 mutation, actually, we can tell. But again, when we talk about BRCA2 mutation, which is the BRCA2 germline or somatic mutation, which is already known oncogenic, they did not consider this BRCA2 RB1 co-deletion or BRCA2 deletion in the single strand. So we believe, as we see that high level of metastatic prostate cancer patients have the BRCA2 RB1 co-deletion, maybe this population, which is considered HRN non-deficient, maybe they have a significant percent of the patient have this co-deletion. To validate this hypothesis, we use the organoid developed from prostate cancer patient. This is the 3 organoid used. They are developed in MSK-CC previously by Icahn group, and MSK-PCA1, PCA2, and PCA3. PCA1 and 3 have co-deletion of BRCA and RB1. You can see that they have a very low amount of the BRCA and RB1 loss, and these loss are the single stranded. MSK-PCA2 have RB1 loss, but this is wild type BRCA. And if we treat with the PARP inhibitor, which is talazoparib, we can see that this talazoparib significantly, even in very low concentration, reduces the proliferation of MSK-PCA1 and PCA3, not PCA2. It means that even the patient who have the single copy loss of BRCA2 and RB1, they may be treated with PARP inhibitor. Even we are thinking if we can move forward and start this treatment very early stage of the disease, even before surgery, like a 3 month of the treatment before prostatectomy, and then do a prostatectomy, that can increase the survival of the node. But again, this need to come to one company should come forward, like Pfizer or somewhere, to sponsor this kind of clinical trial. We are talking with the urologist and geo-oncologist our department. So second, the point is like that we can detect a mutation which is happening early stage of the disease and actually drive the disease progression from indolent to metastatic prostate cancer. And we can treat this particular subtype of disease early stage with PARP inhibitor, which is already approved by FDA for the later stage of the disease. I'm going to this is the summary of our study, which is elegantly summarized by Karen Kunsen's team. What we found that loss of BRCA2 and RB1 induces the changes of cellular morphology, castration resistance, EMC transcription factor upregulation, and the clinical correlate with the prostate cancer progression-free survival, changes of the FGA of action all that, and enrich in the metastatic disease. The question is still remain. The first question when we talk about prostate cancer is, why BRCA2 loss even alone is sufficient to castration resistance? What we see in our experimental study, if we knock out BRCA2 with a CRISPR in LN capsule, which is a castration sensitive or androgen receptor targeted therapy sensitive prostate cancer cell line, it is actually getting resistant even a loss of single gene. Importantly, when we analyze the BRCA2 androgen receptor signaling pathway, which is the major effector of the androgen receptor gene is PSA, we found that BRCA2 knockout cells have a very low PSA expression, but there is another gene called PSMA, prostate-specific membrane antigen, which is the negatively regulated by androgen signaling pathway, which is enriched in BRCA2 knockout cells compared to the BRCA2 wild-type cells. So somehow BRCA2 regulate the androgen receptor signaling pathway, not only doing a DNA damage pathway, it's have a different function than known canonical DNA damage pathway, and it regulate the hormone signaling pathway. That's why maybe the talazoparib trial was so successful, because they combined the talazoparib, which is PARP inhibitor, which is androgen receptor blocker, that inducing somehow the BRCA kind of phenotype and regulate, means like increase the patient survival. So what going on here? We check this PSMA expression, because the PSMA currently is very hot and new topic, we can tell, because of PSMA targeted radiotherapy is currently approved by FDA. So we checked that PSMA expression by histopathology in the cells which is developed, and found that most of the BRCA knockout cells express a high amount of PSMA in cell surface. It is the cell surface receptor. And we collaborate with Dr. Kishore Pillassetty's team in MSK Sethi, who is a nuclear medicine person. And we found that if we can inject the PSMA positive cells in the mouse, and we can image the tumors by non-invasive imaging. So this kind of PSMA imaging, which is actually PSMA PET imaging, which is approved by FDA, can use as a detect maybe the BRCA2 mutation. Maybe we need to more study PSMA mutations are associated, PSMA expression associated with the BRCA2 mutation or not. We need some retrospective study. But important part in like that, how it is happen. So we start reading any kind of data from the previous studies present or not. What we found that there is an old study from Inder Verma's group. They saw that BRCA actually directly interact with androgen receptor in an experimental setting of breast cancer. We repeat this experiment in prostate cancer. We found that if we do the immunoprecipitation experiment, BRCA actually directly interact with prostate androgen receptor, and that means loss of BRCA changes the androgen receptor signaling pathway. Means it's not only doing a DNA damage pathway, but also regulating the androgen receptor signaling pathway. As a result, PSMA going up and the cell become castration resistant. This is our project proposal, which is my lab is working. As you can see that what we proposing that here, and what we hypothesize that loss of BRCA2 mutation, changing the whole system of the androgen receptor. As a result, it induces the PSMA expression. So we can target it, this BRCA2 mutant cell line have high PSMA expression, either by PSMA targeted radiotherapy, which just a year ago approved by in USA, and combined with the PARP inhibitor. So we are trying to validate this hypothesis using this BRCA2 cell line, which is developed in our lab and using a preclinical mouse model system. So conclusion of this part, we can find a fraction of the prostate cancer have this co-deletion, and this co-deletion fraction, and we proposing two different treatment. One is PARP inhibitor alone, or PARP inhibitor combination therapy, to treat this prostate cancer, this lethal subtype of prostate cancer. I'm with the second part of my study, which is want to discuss, because of in the genome, when we look, what I tell that there is a missing part is not sequenced with the Y chromosome. Y chromosome, or the male chromosome, is one of the smallest chromosome of the human body, but it's a very unique. Because of this is very unique, because this chromosome is like have less number of gene in the human body, means only there is a 40 gene present in the Y chromosome, which is actually, and rest of the Y chromosome repetitive sequence. But loss of Y chromosome associated with cancer is known for a long time, particularly from karyotypic study, but functional significance is never known. Our group first time showed that there is a gene called KDM5D loss in Y chromosome in the human prostate cancer sample can promote the cancer progression. So there is another subtype of the cancer may be found in the localized disease, which may have loss of Y chromosome. So cartoon I can always show that there is like if you compare a mouse with the human, mouse a smaller size, but if you compare the mouse Y chromosome of the human, mouse Y chromosomes are like a humongous, like a 700 gene present in the mouse Y chromosome. And whereas a human have only 45 gene in the Y chromosome. So means somehow evolutionary growth for the human, like mammals to like in the primate like human, and we found loss of several genes of the Y chromosome. Even Y chromosome genes are very different. Even our close relative of chimpanzee, the human and chimpanzee Y chromosome is very different. So human Y chromosome is very unique in structure. That's why you need to do more research. Last several years, they saw that the loss of Y chromosome, particularly not in the somatic tissue but blood, germline associated with the disease pathobiology, particularly in cancer, even for the cardiac disease. So there is significant connection between Y chromosome loss and a cardiac attack in the male, and maybe we don't know even the functional significance. Yes, because of the problem is Y chromosome is not able to sequence, because of Y chromosome have several repetitive sequence called paralog, and that's why Y chromosome is in the genetic studies, Y chromosome cannot be sequenced yet. And the expression of the Y chromosome gene in the mRNA level is very low, because of all the genes human body have 2 copies, whereas the Y chromosome only have the 1 copy. But importantly, we also show that the protein expression is not that so low, but we don't have the good antibody to check the every protein by immunohistochemistry for Y chromosome gene. So the mRNA expression, when you do the RNA sequencing study or single cell sequencing study, most of the study will miss the Y chromosome in there. But the last 3 or 4 months this year is very interesting for the Y chromosome people working. There is a 4 big paper came in the in recently in the big journals, and they show that by short I can say that Y chromosome loss is associated with the cancer progression, and the low Y chromosome, the patients who have the loss of Y chromosome, they may have experienced a bad or very aggressive type of cancer. So how it is happened? So we see that first time what I say that when we analyze the Y chromosome gene expression in the prostate cancer samples, which is the prostate cancer cell line, we found that most of the aggressive C42, which is the castration resistant or PC3M, very aggressive prostate cancer cells, have very low amount of Y chromosome. We manipulate the Y chromosome in LNCAP cells, which is the castration sensitive prostate cancer, and we found that when not Y chromosome, we can KDM5D in prostate, these LNCAP cells, we found that loss of KDM5D inducing cellular invasion as well as tumor growth. This experiment was performed by Dr. Kazumasu Komura, my colleague, currently as an associate professor in Osaka Medical School in Japan. But this is the first time which we show that there's a functional significance of Y chromosome gene loss in prostate cancer progression. Harisa is a postdoc joining our group just before COVID. She want to see that, okay, she told that we know the Y chromosome loss, but is it true? Can we visualize the Y chromosome? Should she, with the help of the MSKCC molecular cytology core facility, she developed the paint or it's a fluorescent in situ vibration based chromosomal paint analysis where we totally colored the X and Y chromosome. We do found this LNCAP cells, which is a castration-sensitive prostate cancer, it have 2 X chromosome and 2 Y chromosome. Whereas the other some aggressive subtype of prostate cancer actually losing the Y chromosome. You can see LNCAP C42, PC3, all cells have the loss of Y chromosome. Indicate that this Y chromosome loss actually common. These cells are developed from male disease, but they still don't have Y chromosome expression. And we also found one of the organoid, which is this is the organoid from MCRPC or very high aggressive tumor. We found this one of the organoid which have the Y chromosome PCA1, which is a CRPC, but one of the NEPC organoid we found there's 2 Y chromosome, but they are fused with the autosome, and the loss of the fusion of the Y chromosome with the autosome associated with the loss of Y chromosome gene expression. You can see that there is not only loss, there is a structural arrangement can be happening in the Y chromosome, which we don't know. So to do this thing, we need to develop a sequencing method to develop Y chromosome sequencing in the human clinical sample or solid tumor tissue as well as blood to find that is the loss of Y chromosome associated with cancer progression, or the patient or the prostate cancer patient who have Y chromosome loss, are particular subtype of the disease which can be need to be a different kind of treatment, which can be more attention. To do so, I collaborate with the genomics core facility in the Mount Sinai, which is my current institute, and we are developing a method called PacBio single molecule real-time long-read sequencing to sequence the Y chromosome in the prostate cancer somatic tissue or in the germline to understand that the loss of Y chromosome is it associated with a particular subtype of the prostate cancer. Together, our data with the genetic subtype, we say that we define 2 genetic subtype of the prostate cancer. Those are present in the early stage of the disease and need a different kind of treatment. I have around 5 to 10 minutes. And last part of the talk here, I want to describe something. How can we manage the castration-resistant prostate cancer, which is, as I mentioned, the castration-resistant means the prostate cancer do not sensitive to anti-androgen therapy. Again, when you start the experiment, we are starting for the clinical samples. So we want to analyze some of the clinical data set where the patient in the localized prostate cancer patient treated with the anti-androgen therapy, and we want to know how they can become resistant, not the castration-resistant prostate cancer patient who are treated with the second-generation anti-androgen. We take the patients which are very early disease, prior to any treatment, they are treated with anti-androgen. Found this thing, there is one of the data set we use, which is data set previously, the clinical trial which previously found from my mentor, Dr. Phil Kantoff, when he was in Dana-Farber, that this patient, actually the prostate cancer patient, advanced localized prostate cancer patient, treated with LNRH, which is the anti-androgen inhibitor Lupron, or along with the second-generation anti-androgen abiraterone, and then all the patients after 24 weeks, they undergo radical prostatectomy. We collaborate with the group of Adam Sorosky, who is currently in NIH, that to understand the genomic difference between the patients who are treated with anti-androgen therapy versus no treatment in localized prostate cancer. We do found the anti-androgen therapy regulate the AR signaling pathway, which we call AR score, but induce a molecule, which is BCL2, which is anti-apoptotic molecule, significantly. Only BCL2, they do not induce other anti-apoptotic molecule, the BCLW or BCLXL. So this is hold the data in the clinical samples, but sometimes clinical samples data we cannot able to reproduce in the experimental setting. So Rahim joined in my team, and he did an amazing experiment in the doing an experiment where he saw that this enzalutamide, which is anti-androgen inhibitor, can induce BCL2 expression in the castration-sensitive prostate cancer cells. The R188, which is androgen, he treated the cells with the synthetic androgen first. We found that BCL2 expression is going down, but when we treated the enzalutamide with the dose-dependent condition, we found this BCL2 expression is significantly going up. The same time, all the AR signaling pathway genes are going down. It means that treatment is working perfectly. That's data support that we can replicate the result for the clinical dataset in our lab in the cell line system. Importantly, when Rahim performed this experiment in a castration-resistant prostate cancer, which means this LNCAT ABL subline is developed from LNCAT and do not respond to castration therapy, we found that this castration-resistant prostate cancer enzalutamide does not induce BCL expression. So regulation of BCL2 by anti-androgen therapy is only happening in castration-sensitive prostate cancer, not in castration-resistant. Even if we analyze this data set from the stand-up to cancer, we put side by side our lab data and the clinical expression sample data. Even in the castration-resistant prostate cancer, treatment of the enzalutamide abiraterone does not induce BCL2 expression, indicating that this regulation of the BCL2 by enzalutamide is actually happen in the CSPC, means castration-sensitive prostate cancer cell, rather than CRPC cells. So this is an early event. Even cells are present in the androgen, doesn't matter. The state of the cells actually regulate response differentially to the treatment. Based on this data, we want to understand that what is the BCL2 expression is doing. Are they just a passenger event, or is a driver event of the metastatic or aggressive disease? So what we did, we take the control cells, which are LNCAP cells, overexpress the BCL2 and treated with the synthetic androgen, perform the androgen signaling path, perform the RNA sequencing to understand the signaling pathway, which is enriched in response to androgen in the BCL2 overexpressed versus non-overexpressed cells. Importantly, we found there is 4 unique pathways enriched in BCL2 overexpressed cells compared to control cells. And look at that, all the pathways which we found, actually nothing associated with androgen receptor signaling, everything associated with the cancer metabolism. So, which is we don't know, we never found this thing, and nobody knows studies of showing that androgen receptor have a role on the cancer metabolic pathway. But we do know metabolic reprogramming may associated with the cancer progression. So this is the first time our data suggests that there is a possibility of AR-BCL2 signaling pathway in BCL2 overexpressed cells regulate the castration-resistant, maybe upregulation of the androgen signaling pathway. The androgen metabolism signaling pathway. The metabolic pathway in the body generally regulated by the PI3K signaling pathway. prostate cancer cell, which is LNCAP or other, have a loss of the P10, and that regulate the metabolic reprogramming on the cancer cells. And we know that the P10 loss is regulate on the metabolic reprogramming. Its metabolic reprogramming happen to the activation of AKT. So we think that is it possible the BCL2 regulate the AKT or PI3K signaling pathway? What we did when we analyzed the Western blot, we found that BCL2 overexpressed cells have the high AKT phosphorylation. Interestingly, at the same time, we found that if we knock down the BCL2 siRNA, there is a downregulation of the AKT phosphorylation in the same cell. It means we saw that BCL2 can regulate AKT signaling pathway that maybe regulate the metabolic reprogramming. On the other hand, when we knock out we just make changes in the AKT PI3 kinase signaling pathway, taking the knockout the P10, we found that P10 loss is inducing BCL2 expression in the mouse model. And if we knock down the P10 also in the prostate cancer cell line, which is 22RBN, we found with the P10 wild type cell also inducing BCL2 expression. It means that AKT signaling pathway also by a feedback loop expressing BCL2 expression. And same thing we found with the AKT inhibitor, which is MK2206, in the same cell line, we found that AR inhibitor-induced BCL2 expression is downregulated by MK2206 AKT inhibitor. Taken together, there is a feedback signaling loop we are first time reporting, which actually regulate between the treatment of AR inhibitor therapy, and this reciprocal feedback signaling loop regulate the both BCL2 and PI3K signaling pathway, and upregulation enrichment of these two signaling pathway may induce castration resistance from the castration sensitive prostate cancer, which is the early stage of the disease. We go forward little. We do the clinical validation. We take the venetoclax and combine with enzalutamide to check the mouse tumor growth. Can it significantly reduce the castration-sensitive prostate cancer tumor growth? We use a mouse tumor model which is called MICCAP because of we want to use a syngenic model due to the presence of expression of the BCL2 in the blood cells. And we found that BCL2 enzalutamide combination significantly reduces the growth of the tumors. We found, you can see that individual tumor growth here, and you can see how nicely they can reduce the tumor growth. We are trying to take this combination therapy for early stage of the disease. Earlier, what we did, we just give only androgen therapy in the localized prostate tumor. We want to give this localized castration-sensitive prostate tumor, this combination therapy. The major burden is like that with venetoclax is a drug, FDA-approved drug, used in the liquid tumor of the leukemias and the blood cancer. But it's a FDA-approved drug, although FDA-approved drug, the company AbbVie do not want to use this drug for any kind of solid tumor clinical trials. So our hand is kind of try. We're trying to, that funding agency, we can use this clinical trial using venetoclax and enzalutamide together for the castration-sensitive prostate cancer, which can delay that castration resistance from the castration-sensitive prostate cancer. So, in summary, we also use this venetoclax and AKT inhibitor for castration-resistant disease, and we found that this venetoclax AKT combination can reduces the cell proliferation even in the castration-resistant disease. So this can be an option for the treatment for castration-resistant prostate cancer. So we combine, if we take my first slide, one of the slide, prostate cancer treatment, we can induce the BCL2 inhibitor for prostate cancer treatment. We can combine BCL2 inhibitor on the ADT, very early stage of the disease, or we can combine the ADT, venetoclax, and PI3K inhibitor, particularly for P10 mutant tumor, even in the early stage or the latter stage of the disease. So using BCL2 inhibitor may be in different combination, in different stage of the disease, may be beneficial for the prostate cancer treatment and can be increase the survival. I know I am spending almost like 45 minutes of my time. I'm not going move forward much, but such kind of research, what we do it, the different aspect of prostate cancer cannot be possible without a group collaborator. I make collaboration with my previous group with Dr. Kantop. Currently, he's a CEO of Convergent Therapeutics. My previous lab mate or my current lab mate, I collaborate with different places, MSKCC, Dana-Farber, MIT, and some other, my current institute, Icahn School of Medicine, to perform my experiment, moving forward my lab. Fortunately, I get supported from several organizations, including NIH and DOD, and particularly Prostate Cancer Foundation. Always thankful to them to support my research. And thank you so much. I'm ready for the question, and I'm happy to answer and discuss about my science. And last, I can say that I'm always open for collaboration. You can reach me anytime, and we can start working in the prostate cancer or any other interesting field associated with cancer biology. Thank you.
Okay, any questions?
I can see question in the chat.
Yeah, I have one. So thank you very much, Dr. Goutam. That was quite impressive and a little bit frightening as well. So when I looked at your CNA data, showing that pretty much every chromosome either has a gain of chromosomes or loss of chromosomes, I appreciate how you selected chromosome 13 and the BRCA2 and everything. But my question is, when you look at some of these, especially the more aggressive tumors, what is the outlook? Does every tumor always have CNA in all chromosomes? Or have you looked at the average number of chromosomes with, CNA as a factor for prognosis and behavior?
Yeah, great question. So I just go back to my previous slide. That will be easy for me to answer. Just go there. Yes. Yes. Let me put it. Okay. So
yeah, that was the one. Yeah.
Yeah. So you can see that it's at this is a combined data set, not an individual patient. Okay.
Yeah.
It's like it's not necessary that all the patients have the chromosome 13q deletion. And as you saw that it's like in metastatic prostate cancer, it might be a 50% patient have this deletion. But other 50% patient maybe have some other deletion here, which is not overlap. So it's a very specific to the patient. So that's why we have to do an individual analysis patient by patient to see that.
Yeah.
This is a whole, but what we do see that this amplification is kind of a random. Yeah. We don't see a very specific. Commonly, we know that a chromosome HP is amplified every time, 8q is amplified every time, which is a MYC. But MYC amplification is not an individual image, it's amplified with several other genes. Yeah. So deletions are kind of a specific, amplification kind of a random. So means we don't know amplification how regulate. It might be some patients have some deletion is associated with some amplification. And that may be a interaction between, when you have a gene is deleted, it's a tumor suppression, or gene is amplified, we consider it an oncogene. But this interaction map is not, we don't have yet. We definitely collaborated with the computational biologists, mathematician to make this interactive map. But yes, if we start from the CNA, we might found something, and that kind of interactive map, when we found, I can design the experiment in the cell line system and think how the interaction between two genes are growing. And that will be a good question about it. That can be give us like the synthetic lethality concept to target this oncogene when the patient have particular kind of genetic mutation.
Yeah, thank you very much. I was just thinking because we know in general that the higher the mutational burden is, the more aggressive an average tumor is. So I'm just thinking that maybe the CNA is just another way of looking at the mutation burden and see how that goes. But thank you very much.
Yeah, that I can just mention one point that we did analysis between that data is not here. We did analysis between mutational burden and CNA in prostate cancer.
Yeah.
Actually, they are not actually overlapping each other. I mean, patients who have very high CNA have low mutation, and those we found very aggressive.
Okay. Oh,
so
interesting. Yeah.
It's a cancer type. Prostate cancer is cancer which is not very mutational driven cancer. It's like a more genetic abnormally driven disease.
Yeah, thank you. I see that Scott's hand is up too. So Scott, sorry.
Hi, Scott.
Hi, Goutam. I really enjoyed that. Thank you very much. I was interested about the part of your talk where you were discussing the co-deletion of BRCA2 and RB1. Can you just elaborate on that genotype a little bit and its clinical course and behavior relative to those tumors that have BRCA2 loss alone or RB1 loss alone?
Okay, so as you can see, this is the data. Sorry, next one. Yeah, this is the localized tumor. The data we have. You can see the BRCA2 tumor alone have very few number. So this is when we analyze. Even I can come to the slide. Okay, let me see here. Okay, so you can see this all the patients which are the BRCA2 RB1 loss. you do not find a patient here, or very less patients who have BRCA2. Maybe some patient here have BRCA2 loss, and BRCA2 loss without RB1 loss is very rare. It's like a outlier kind of thing. There is a patient have RB1 loss and they don't have BRCA2 loss. That patient actually doing better. Actually, they are not much difference from the localized which don't have any deletion. So we believe the whole two loss actually have better effect. So when we knock down this one, if you can see that this is like a totally changes of the gene expression pattern when a co-loss. Also, you can see here the AMT kind of phenotype where we make the co-loss, they actually have these changes. So it means this co-loss making some have some effect which is considered we completely don't know, but still how this happened, that is our major research. We are doing more things, but we found one molecule which is the DNA damage, one is the cell cycle. When the cell cycle regulatory checkpoint is over, the DNA damage molecule actually lost, supposed to be lowering down the cell cycle progression, but it's not happening because of the cell cycle gene is getting lost. And there also have some transcriptional regulator affecting directly to the anti-androgen signaling pathway. I do like to mention that there is several data from prostate cancer patient, particularly Karen Kunsen group. They show that RB1 directly interact with androgen receptor, and RB1 loss in later stage of prostate cancer, which actually have BRCA loss, they may regulate the cancer metabolic pathway. So it's a complex signaling pathway. We don't know completely because we found this thing like 3-4 years ago, this mutation. We're still working. One big problem for us is like using the right model. We cannot use a mouse model because in mouse, BRCA and RB1 is not in the same chromosome. Mouse is 114 and 5. We talked with some of the group and they're trying to reproduce the result, and human and mouse result is completely different. But I do want to show you this, mention this data, which is support my hypothesis completely true, because you see, this is the patient from the Spanish Cancer Center. This patient have germline BRCA mucaria, and this patient with the green have germline BRCA peria plus BRCA co-deletion. So that patients are doing really bad. And from this David Einstein group, those patients have this co-loss, even the single copy or double copy, they are more immunogenic. So this BRCA RB1 loss doing multiple important things. One of the important things we describe is the cellular plasticity, but also in the tumor microenvironment, they increasing the immunogenicity. But how? We still don't know.
Cool. Well, we can talk some more about it in a little while. I'll meet with you later this morning.
Yeah, definitely. Looking forward.
Look forward to it.
I think Andy has a question, right?
Yeah, thanks, Sid. Hopefully it's not too loud, Goutam. Thanks. Great talk. I'm going to try and link a couple of things that you actually presented disparately, and it goes back to actually work that I did, like we were talking about last night, as a BRCA1, BRCA2 scientist back in the day. One of the things that our lab showed was that PI3 kinase AKT activity was required for BRCA1 expression, and then Jose Baselga's lab subsequently showed that inhibiting PI3 kinase would actually sensitize breast cancer cells to PARP inhibitors. I'm wondering, again, seeing that you're seeing some of the similar things with the BCL2 story and the castration resistance and the PI3 kinase role in there, have you looked to see what PI3K AKT activity looks like when you compare your BRCA2 RB1 co-deletion models versus your BRCA2 only versus your wild type? Do you see differences in PI3K AKT signaling across that genomic spectrum?
Yes, that's a very good question. And so, sorry, I didn't show the data here because I cannot save the data. I do take this thing. So after publishing this paper in the first paper in BRCA2 RB1 co-deletion, we do a follow-up study to cell signaling pathway which are regulated in the under the loss of BRCA2 particularly. What we do see in LNCAP there is a significant enrichment of AKT signaling pathway, activation on AKT. Surprisingly, we also see there is a significant upregulation of phospho-SRC tyrosine kinase pathway. But when we treat these BRCA knockout cells with the SRC inhibitor and PI3K inhibitor, we do not find PI3K inhibitor have any kind of significant cell growth inhibition, whereas the SRC inhibitor loss, we found there is a significant amount of cell growth inhibition. So we do see according to same like what is the Baselgas group, so that became BRCA1 loss, and also like a PI3 activation. But what in contrast to their study, our study show that even there is an activation of the AKT signaling pathway, but these cells are not dependent on AKT signaling pathway. Means even this is a very preliminary data, but when we treat the cells with the AKT inhibitor, they do not respond so well. But definitely they are hyperactivated or hypersensitive with the SRC signaling. So there is a downstream signaling pathway definitely activated, but AKT, even if activated, it might not have any significant role yet. But okay, we have to do more other study to validate this analysis. But I can mention one point. Some of our colleagues did an experiment in the PTEN knockout mouse. What they saw that PTEN knockout mouse have some kind of a DNA damage response, particularly activation of CHEK2. BRCA, I checked the BRCA expression in PTEN knockout mouse. Actually, BRCA expression is high, going up in PTEN loss. So those data are not published yet, but yes, we do try to connect, because of this is two major projects in my lab, PI3 kinase and the BRCA2. We try to connect each other, but still not it's successful yet.
Okay, we have Dr. Zhao.
Yeah.
Great talk. I really enjoyed it. So I have a question regarding your Y chromosome loss in the prostate cancers. So in the normal situation, so female has two X chromosomes, so they have a random inactivation to balance the X chromosome compared to the male. Do you know any mechanism the female gain some to compensate that they don't have Y chromosome? Which kind of mechanism they compensated for that? And my second question is also, Y chromosome loss is not specific for prostate cancer. For all the kind of cancer and in the male patient, you often can see the Y chromosome lost. So how do you comment on that?
Great question. First thing, the compensation of the X chromosome by Y in female is a very important question. And unfortunately, I don't think any research is there going on, because of, see about the most of the paralog genes of the Y chromosome, which is KDM5D. We talk about KDM5C present in the X chromosome. They have kind of similar function or kind of dissimilar function. But we don't know. I mean, we do see in prostate cancer, like there is some patient lost one Y chromosome, gain one X chromosome. But I don't know this X chromosome function as a female X chromosome or not. It should be, I mean, that's a, I mean, as you can see, the cancer and the sex chromosomes are not a very popular field to work. Not many people work on the role of sex chromosome in the cancer. So we don't know. But there is a study from the UK Biobank, what they show that some of the regions are conserved in Y chromosome, also in X chromosome, okay? That region, if it's deleted in the blood, they are actually associated with poor prognosis. So it's like that, I mean, we don't know yet. Really, there is no much study there, so that I don't want to comment anything. But yes, there is definitely some function in this sex chromosomal interaction in cancer, because of particularly those cancer associated with the what we call like a hormone-dependent cancer, like breast cancer, ovarian cancer, and prostate cancer, maybe. But we are not there to answer definite answer the question that why there is a compensation of the Y chromosome in the loss in the Y chromosome null condition, like a female cancer, which is the X chromosome compensation, 2X chromosome, how compensate the Y chromosome function? Really, we don't know the answer. The second part is like that, yeah, Y chromosome loss is very common in all male cancer. So interestingly, as you know that males suffer more cancer, I mean, males have the chances of getting more cancer than female, and males have 40% more chances to die by cancer than a female. So, and now if we talk about Y chromosome loss, we talk about Y chromosome loss in the somatic tissue and germline. All older male, like after 40 or 45, they have a mosaic loss of Y chromosome in their blood. Is this associated with cancer? According to the UK cancer research, yes. Is this associated with a particular subtype of the cancer? We don't know yet. What is the recent result from the Sinai? They show that loss of Y chromosome in the bladder cancer is associated with a very poor prognosis. Importantly, bladder cancer patients have also associated with the high smoking rate, and we know that the smoking is actually associated with the loss of Y chromosome in the blood. So it means we don't know a total connection between Y chromosome loss, smoking, and cancer. We need to do in bladder cancer. we do see kidney cell cancer have the loss of Y chromosome associated with the bad prognosis. Importantly, the data from the DPNO group in the, I think so that's like a colorectal cancer, they show that loss of Y chromosome actually good prognosis for the colorectal cancer patient. So somehow Y chromosome presence is associated with the good prognosis. So we are not there yet. So if the Y chromosome research, not many groups did it. We are the only group for doing the prostate cancer. What data we saw that Y chromosome loss may be somatic tissue, bad prognosis, some cancer like in prostate cancer, in prostate cancer, in bladder cancer, maybe kidney cancer. We see the data from the DPNO group. It may be good prognosis for colorectal cancer. But again, DPNO group use a mouse model completely. As I mentioned in my talk, mouse and human Y chromosome is very different. But yes, overall, if you see the loss of Y chromosome in the blood, that associated with the cancer. And one thing I want to mention that in the recent study, you familiar with a term called clonal hematopoiesis, which is the accumulation of the mutation in the blood, random accumulation. We do see there is a strong connection with the loss of Y chromosome and clonal hematopoiesis. And there's some studies from the Memorial group. They show that clonal hematopoiesis is actually associated with the subdue immune response as the result of the activation progression of the bad aggressive cancer.
Great, thank you.
Thank you.
Well, thank you, Goutam. It was a great talk. And I know you're going to be meeting a lot of people. So thank you again. It was a very, very great talk.
Thank you so much.